today's class. Today we are going to learn about cell. This class is based on Ncert textbook, uh, and today we are dealing with chapter 8, cell, unit of life. The topics we are going to discuss today deals with the definition of cell. Then we will see the cell theory. Moving on to the overview of a cell, that is the basic characteristics of a cell. Then we will see the difference between prokaryotic cell and a eukaryotic cell. Finally, we will look into the bacterial cell, which is an example for a prokaryote. So let us start. So there are several ways one can define cell, and a cell can be considered as the smallest unit that have the ability to live its own. So for example, if we consider the unicellular organism bacteria, let us say take the example of an E. coli. It lives in as a single celled organism, which means a single cell of E. coli can survive on its own. That particular single cell has the ability to carry out all the metabolic and other chemical reactions that are required for any life form to sustain life. So this is what is meant by a cell is the smallest unit that can live on its own. In addition to this, uh, inside a cell there are several cell organelles. A cell is surrounded by a plasma membrane and so on. The point is that cell can survive on its own, being it unicellular or multicellular organism, it can perform all the functions. The thing is that inside a cell, even though there are many cell organelles, but not the cell organelles inside the cell can survive on its own. Which means it requires the cytoplasm, it requires the delimiting membrane, and coordination of all other cell organelles for a cell to function. Not a single organelle can carry out its function on its own. And when we consider the cell sample, E. coli cell, it can multiply, it can undergo cell division independently, and that is not the case with multicellular organism. Multicellular organism can and also multicellular organisms can also um, undergo cell division. They can replicate. The cells have the ability to divide. And when we say cell is the fundamental unit of life, it simply means that cell is the basic unit in which all the necessary metabolic, chemical pathways and reactions are carried out, and these reactions are essential for sustaining life. And cell, it is the basic unit in which all the necessary actions, activities are taken place, and that is what we meant by saying cell is the fundamental unit of life. And of course, cell is the structural and functional unit of life, which means whether we look into the unicellular or multicellular organism. In the case of multicellular organism, there are different types of cells, and that makes up the tissues. And finally, different tissues give rise to organ, and the complexity uh, increases at each level. So whether it is unicellular or multicellular, cell is the basic unit in which all the necessary activities are taken place, and as a result of the activities of the cells, the tissues can perform well, and so does the organs and so on. So this is the idea of a cell. And now we are going to see the important studies or discoveries that were made by various scientists in the field of cell biology that ultimately contributed to the formulation of cell theory. And here we are going to look into the some of the selected scientists and their works, and we will see how it has led to the formation of cell theory. This is only to brush up the previous knowledge. Today we will see only some of the important uh, studies conducted by various scientists, and we will start with Robert Hooke. He was the first scientist who coined the term cells. and he was working with cork, which is a plant cell, basically. And when he looked the specimen under microscope, he saw certain kind of box-shaped structures which resembled or which reminded him of the cells or rooms that one can see in monasteries during that period. Therefore, he coined the term cells, or he used the term cells to define the structure that he examined under the microscope. And this was the first uh, discovery that was made in cell biology. And he basically looked at the dead cells of the cork of plant cells. Now we'll see Anton van Leeuwenhoek. In 1670, he was able to see live cells. That is, he looked into the sperms of animals, he collected the specimen, and he observed the cells moving under microscope. He was the first person to see a live cell. Whereas Robert Hooke was the first person to see the dead cells, and he coined the term cells. And we will see the next one. In 1831, Robert Brown he discovered nucleus. Uh, Due to the advancements in science and technology, there were improved instruments. As a result, Robert Brown was able to discover a cell organelle that is more or less centrally located, and he discovered nucleus. And finally, Sheldon and Shaw in 1839 they hypothesized cell theory. They formulated it, and they put forward the theory, cell theory, which we will see later. These are the some of the works that led to the contribution of cell biology in general, as well as to the formulation of cell theory. Cell theory was formulated by Matthias Sheldon and Theodor Schwann in 1839, and later their work was modified by Rudolph. We will see the what was cell theory and the contribution of each of these biologists. Matthias Sheldon, he exclusively worked on plant cells. He observed many different kinds of plant cells, and he came to the conclusion that plants have different kinds of cells, and these different kinds of cells give rise to tissues that one can see in plants. So this was the formulation or the hypothesis made by Matthias Sheldon after examining plant cells. His work was exclusively based on plant specimen. Now we will see the contributions of Theodor Schwann. He was basically into animal cells. He studied several animal cells, and he came to the conclusion that there is an outer layer to the cell, and he found that that plasma membrane surrounds cell. His studies on plants and as well as animal cells came to the conclusion that cell wall is unique to plants, and cell wall is absent in animals. One cannot see cell wall surrounding the plasma membrane or the cell membrane of the cells seen in animals, whereas it is clearly evident in the case of plant cells. And later on, he came to the conclusion that animals and plant bodies are composed of cells and products of cells. So basically, whatever the living organism is, their bodies are made up of cells. These were the conclusions of Theodor Schwann after conducting his extensive research in the area of cell biology. We can say that the combined efforts of Matthias Sheldon and Theodor Schwann came to be known as cell theory, but there was a drawback in the cell theory proposed by Sheldon and Schwann. This is because there was no studies conducted on the cell division, that is how cells divide. Nobody knew during that time, during 1839. But later on, due to the advancements in science and technology, a scientist called Rudolph. In 1855, he was successful in explaining how the cells divide, and therefore he made up the whatever drawbacks was in the cell theory proposed by Sheldon and Shaw, and he modified their work. He was able to explain how cell divided and how a new cell arise from the existing cell, and therefore the cell theory was modified. This is the cell theory that is now universally accepted. States that all living organisms are composed of cells and products of cells, and also all cells arise from pre-existing cells. So this is what cell theory is all about. There are many living organisms, and whether it is unicellular or multicellular, their body is made up of cell. So cell is the functional and the structural unit of life. This is fundamental unit of life, and all the living organisms, their bodies are composed of cells or the products that arise from the cells as a result of various reactions, and the cells divide so that the new cells arise from the pre-existing cells. Cell division occurs, and this is how the cells work in body to maintain the life. And now we will see the overview of a cell. So cytology is the branch of science that deals with the study of a cell. It deals with the composition of cell, the structure, the function, the morphology, the form, the different types of any cell. And that is what cytology deals with. So, coming to the overview of a cell, we know that every living organism is composed of cells. Whether it is unicellular or multicellular, cell is what makes that living organism to survive. Taking in the example of unicellular organism, there are prokaryotes and eukaryotic organisms that are unicellular. The most common prokaryotic unicellular that we all know is the bacteria. Whether it is eukaryote or prokaryote, the in unicellular organism, it is that single cell that performs all the essential functions of life, and it can exist independently and can undergo cell division to form the next generation, the case of unicellular. When we are looking into the multicellular organisms, there are millions and millions of cells, and in an organism, say for example man, there are different cells give rise to tissues, and different tissues give rise to an organ. Different organs, they function, they comes under an organ system, and this different organ system coordinate the various activities in a body or in an organism. This is how uh, the life exists, or this is how a multicellular organism live. So basically, the single unit, that is the cell, acts as the fundamental unit of life, whether it is unicellular or multicellular. And we know that cells comes in different shapes, size, and therefore the activities a cell perform is also different. Now, if we consider the size of the cell, there is the smallest and the largest cell. The smallest cell is mycoplasma, which is a prokaryotic cell, but it lacks cell wall. And the size of mycoplasma is zero point 0.3 micrometer, whereas the largest cell is the egg cell of the ostrich bird. When we consider the human body, the longest cell in human body is the nerve cell, while the smallest cell is the sperm cells. Now we'll come to the shape and the function of the cell, how the shape determines the function of the cell, or how the shape favors the cell to perform a particular function. Take for example RBCs, we know that it is disc shaped, or it is biconcave. This is because the function of RBC is to carry oxygen, carbon dioxide, and transport various other nutrients, etc. So as to make the transportation easier for the RBCs, the shape of RBCs are either disc shaped or biconcave. Now, when we look into the WBCs, that is the white blood cells, which provides immunity to our body. They are more or less irregular in shape or amoeboid in shape. Particular shape allows WBCs to engulf pathogens easily. And this particular cell, that is, we can see in the uh, epithelium of the intestine, small intestine. It is a long and narrow. This is to increase the absorption of nutrients from small intestine. And this is the relation between the shape of the cell and the function it performs. Every cell has a delimiting boundary, which means that no two cells can collide. They do not club into each other, they exist independently. Even though there is communication between cells, one cell will not collide into the other cell. There is a limiting membrane or a boundary. So, in the case of plants, we can see cell wall. In bacteria, also cell wall is present. In the case of animal, the delimiting boundary is the plasma membrane or the cell membrane, and the only prokaryote that lacks cell wall is a mycoplasma, and it has the cell membrane as a delimiting boundary. And when we see into the cell, inside of a cell there is cytoplasm. It is a fluid matrix. Cell organelles are seen in here, and majority of the cell activities takes place in cytoplasm. Now we'll see the differences between prokaryotic cell and a eukaryotic cell. Prokaryotic cells are smaller when compared to eukaryotic cell. For example, if we consider a bacterial cell, that of an animal cell, when these are compared, when the size of these cells are compared, prokaryotic cells appear smaller than the eukaryotic cell. And when it comes to the cell organelles, we cannot find any cell organelles that are membrane bound. While in the case of eukaryotic cell, there are many cell organelles that are bounded by a membrane. For example, mitochondria, Golgi complex, endoplasmic reticulum, etc. Now, if we look into the genetic material of bacteria or any prokaryotic cell, we can see that the genetic material is naked, which means that the genetic material is not situated inside a nucleus. There is no envelope or membrane that surrounds the genetic material, and there is no true nucleus. But in the case of a eukaryotic cell, we can see there is a cell organelle called nucleus, inside which there is genetic material, and therefore eukaryotes have a true nucleus. If we consider the plasmids, generally plasmids are present in prokaryotic cells that gives them antibiotic resistance, and these plasmids are made using our DNA technology for several purposes. While in eukaryotic cell, we cannot find any plasmids. Now, if we consider the mesosomes, these are present in prokaryotes, but mesosomes are absent in eukaryotic cell. And like I already mentioned, there are several cell organelles that are present in eukaryotic cell but absent in a prokaryote. Now, if we consider the ribosomes of prokaryote and eukaryote, 70S is the ribosome of prokaryote, while 80S is the ribosome of eukaryotic cell. Here S stands for Sedberg units, which is the sedimentation coefficient, and the ribosomal subunits are 50S and 30S in the case of prokaryote, which means 50S is the larger unit subunit and 30S is the smaller subunit, and together larger subunit and smaller subunit give rise to 70S subunit in the case of prokaryote. In eukaryotes, 60S and 40S are the larger and the smaller subunits respectively, which give rise to 80S ribosome. These are the differences between prokaryotic cell and eukaryotic cell. And now we will see prokaryotic cells, the general characteristics, features, and the like of a prokaryotic cell. Like we already mentioned, prokaryotic cells are in general smaller than the eukaryotic cell. Here we can see the picture of bacterial cell as well as an animal cell. The next important feature of a prokaryotic cell is that they can multiply more rapidly than any other eukaryotic cell, which means that if we consider the example of a bacteria, say for example E. coli, it takes almost 30 minutes for an E. coli cell to divide to give rise to a new E. coli cell. While in the case of any eukaryotic animal, typically takes more than 24 hours for a cell to divide. So, when it comes to the rate of cell division, bacterial cells or like prokaryotic cells in generally, they can replicate more easily, they can multiply more rapidly than any eukaryotic cells. And these prokaryotes, they come in various shapes and size. To cite certain examples of prokaryotes, we can say bacteria, blue-green algae, mycoplasma, and PPLO. PPLO stands for pleuropneumonia-like organisms. In general, if you take any prokaryotic cell, we can see a cell wall that surrounds the cell membrane. But this is not seen in the case of mycoplasma. Mycoplasma is also a prokaryote, but it is the only prokaryote which lacks the cell wall and it is a smaller cell. The next important feature of a prokaryote is that they lack true nucleus, which means genetic material is naked, it is not covered by any membrane bound organelle. In addition to this, we can see an extra chromosomal genetic material called plasmids, that is, it is present in the cytoplasm. Along with, we can see a cell organelle that is mesosome. And the another important thing that we can see in prokaryotic cell is a ribosome. Every cell needs ribosome because it is the organelle carry out the process of protein synthesis. Without protein synthesis, a cell cannot survive. Therefore, every cell has ribosomes. But if we consider other cell organelles, it typically in any prokaryotic cell, the cell organelles like mitochondria, Golgi body, endoplasmic reticulum, etc., are generally absent. And these cell organelles are membrane-bound cell organelles. Now we will see the structure of a bacterial cell. We are going to study the structure of a bacterial cell, bacteria which is a prokaryote, in detail. And before that, we will see the some basic things about bacteria. Bacteria, like I said, is a prokaryote, and it comes in different shapes and forms. These are the four basic shapes of bacterial cell. That is the bacillus, coccus, spirillum, and vibrio. If we consider bacilli, that it appears in rod shape. That is, for example, we can say the E. coli. It is a bacilli, which means it has a rod-shaped cell. And if you consider streptococcus, these are spherical in shape, and therefore the name cocci. Then we have the spiral. If the bacteria appear as spiral in shape, it is called spirillum. And if the bacterial cell is resembling that of a comma shaped, then the bacteria is called vibrio. And for example, we can say Vibrio cholerae. The causative organism of cholera is comma shaped. These are the basic shapes of a bacterial cell. Now, if we consider um, any bacterial cell, irrespective of its shape, whether it is a rod, cocci, spirillum, or vibrio, or comma shaped, we can see that a bacterial cell has several structures seen outside the cytoplasm and inside the cytoplasm. For example, if we consider the cell envelope that is seen outside the cytoplasm, cell envelope is made up of three layers: glycocalyx, cell wall, and plasma membrane. These three layers make up the cell envelope. In addition to this, we can see flagella flagella and pili and fimbriae in some cases of bacteria. These are the structures that are typically seen outside cytoplasm, whereas there are several structures that are seen inside the cytoplasm. For example, like we already mentioned, mesosome, ribosome, and inclusion bodies, genomic DNA, and plasmid. And Now we are going to see the structures that are seen outside the cytoplasm. Cell envelope consists of three layers. The outermost is the glycocalyx, the middle one being the cell wall, and the innermost is the cell membrane. Now, if you consider this glycocalyx, its composition is different in different bacterial species. And as such, due to its difference in composition, these bacterial cells they respond differentially to gram staining. It's a procedure used in microbiology, and as a result of gram staining, if the bacterial cells they take up the color of stain, if the bacterial cells appear purple, we call that bacteria as gram positive bacteria. Or if the bacteria appear as pink, then we call that bacteria as gram negative bacteria. So that is about the glycocalyx, and due to the changes in the composition of glycocalyx, there is different types of bacteria, that is a gram positive and gram negative. If in certain cases the glycocalyx is loosely packed, then glycocalyx is termed as slime layer. So if in any bacterial cell the glycocalyx is loosely attached or loosely packed, then that layer is came to be known as slime layer. If in the case of certain other bacteria, if the glycocalyx is thick and tough, if it is thickly packed and it has a tough texture, then the glycocalyx is considered as capsule in that case. The next second layer that comes in the cell envelope is the cell wall. Cell wall, every bacteria has a cell wall and it surrounds the cell membrane. Its cell wall determines the shape of cell, and it provides a support to the cell bacterial cell. It also prevents the bursting of the bacterial cell. This is because bacteria they live in extreme conditions, and cell wall also serves the purpose of preventing the cell from bursting when the cell has to survive in any extreme condition. The third layer that comes in the cell envelope is the plasma membrane. Plasma membrane, it is a semi-permeable membrane. It does not allow every molecules to pass through into the cytoplasm. It is the layer that it interacts with the environment. And typically, the plasma membrane is more or less structurally similar to the eukaryotic plasma membrane. Now, when we look into the plasma membrane, we can see that there is a structure that extends from plasma membrane into the cytoplasm, and these are called mesosomes. In some cases, in certain bacteria, we can see chromatophores. These are the uh, pigment containing structures in bacteria, in some bacteria. These are also the extensions of plasma membrane. The next structure that we can see uh, outside cytoplasm is the flagellum. Flagellum is usually seen as an extension of cell wall. It is thin, it appears as a filament, and the function of flagellum is to provide motility to bacteria. If the bacteria is motile, that is, it's able to move from one place to another on its own, then that is made possible by the presence of flagellum. Typically, flagellum has three parts: filament, hook, and basal body. The next structure is pili and fimbriae. These are seen as surface structures, and the typical function is attachment. It provides a means of attachment to host cell or to any environment. When we consider pili, it is longer than fimbriae. Pili is a long tube made of a certain protein, whereas fimbriae it appears as small fibers, bristle-like fibers. In this pili, it has a function in conjugation, that is when bacteria undergoes sexual reproduction. The pili act as a means to attach to the recipient bacterial cell to transfer the genetic material from the donor to the recipient cell. And fimbrate helps to attach the bacteria to certain environment conditions like rock or to holding onto some host cell. So that's about the structures outside the cytoplasm. Now we will see the structures that are seen inside the cytoplasm. Like we already mentioned, there is mesosomes. If you look into the diagram, we can see that cell wall surrounds the plasma membrane, and the plasma membrane from that an extension is there is an extension that's coming into the cytoplasm, and these are called mesosomes. Mesosomes can exist as vesicles or tubules, or in some cases as lamellae. And the function of mesosome is that it has a certain role in cell wall formation. It helps in DNA replication, and it has a role in respiration as well as secretory process. Since it has a role in respiration, sometimes mesosomes are considered as mitochondria of bacterial cell. Since mitochondria is associated with respiration and energy production in eukaryotes, similarly, in some cases, mesosomes are considered as mitochondria of bacterial cell. The next important structure that we can see inside the bacterial cell is the ribosomes. Ribosomes are cell organelles. These cell organelles are not membrane bound, and if you look at the structure of a prokaryotic ribosome, it is 70S. It has a larger subunit as well as a smaller subunit. 50S is the larger, while 30S is the smaller subunit. And we can say ribosomes are sometimes associated with plasma membrane, and it is the site where protein synthesis occurs. And in case of bacteria, we can see that a single mRNA strand is translated by several ribosomes at the same time, and this gives rise to the structure called polyribosomes or polysomes. The function of ribosomes is nothing but to translate the code in mRNA into proteins. The next structure we can see inside the cytoplasm is the inclusion bodies. Inclusion bodies are reserved materials like nutrients. These nutrients are reserved inside the cytoplasm of the bacteria just in case if the bacteria ran out of the nutrients, or if it has to live in an environment where these nutrients are scarce to find. These inclusion bodies kind of preserve these nutrients. For example, phosphate granules, glycogen granules. In addition to this, in certain bacteria, for example, like especially in blue, green, purple, and green photosynthetic bacteria, one can see gas vacuoles. So, gas vacuoles, phosphate granules, glycogen granules, etc. These are all considered as inclusion bodies, and these are seen in cytoplasm. Like gas vacuoles are especially seen in blue, green bacteria, purple, and green photosynthetic bacteria. The next structure we can see inside the cytoplasm is the genomic DNA. Like already mentioned, the bacterial DNA or the bacterial genetic material is naked. There is no structure that is surrounding the genetic material. Therefore, there is no well-defined nucleus in the case of prokaryotes bacteria, and the genetic material is literally naked inside the cytoplasm. Along with that, we can see plasmid. A plasmid is generally smaller than DNA. It is the extra chromosomal genetic material. It also contains certain genes that provides certain special features to the bacteria. For example, uh, antibiotic resistance. In some cases, if we are given an antibiotic for a particular illness, and if the antibiotic is not working and we are not cured of that particular disease, that may be uh, due to the presence of the plasmid which provide antibiotic resistance to that bacteria. So plasmid has several other applications in our DNA technology as well. So that is all about the prokaryotic cell. I hope this class was helpful for you and thank you for watching this video.